Hello, everyone. This is Dr. Tooba Mahboob from UCSI University, and the topic of my today's presentation is anti-acanthamoebic activity of plant-derived compounds entrapped in polymeric nanoparticles. So we'll start our presentation with the parasite. Basically, Acanthamoeba comes under the protozoa class of parasite, and then it is classified as a waterborne parasite along with crypto and giardia. So acanthamoeba, basically they are the free-living amoebas, and they are considered as an opportunistic pathogenic parasite. They are ubiquitous in nature, so they can be found in soil, air, every kind of water like drinking water, sanitary, dental, and so on. They're highly pathogenic. Basically, it can cause a sight-threatening disease known as keratitis in immunocompetent individuals, whereby they can lead to a fatal disease known as amoebic granulomatous encephalitis, particularly in immunocompromised patients. So moving towards the life cycle of acanthamoeba, basically it exists into two main stages. One is the vegetative infective stage of parasite known as the trophozoite stage. It's a single wall structure, irregular shape structure, and they also have acanthopodia, whereby under harsh conditions such as high temperature, high pH, high chemical pressure, it can transform itself into the more resistant and the resilient stage that is called as the cis stage of the parasite. It's a double wall structure and they can be found in various shapes like round, triangular, or even star shaped. So on the basis of morphology, basically, acanthamoeba, pathogenic acanthamoeba species have been categorized into three main groups, group 1, 2, and 3. Acanthamoeba triangularis basically belongs to the acanthamoeba group 2 along with other pathogenic amoebas like Acanthamoeba castellani and Acanthamoeba polyphaga. So basically the transmission of acanthamoeba into human being can be through various ways. One is it can be enter into the human body through eyes, even through nose, and then it can pass to the lower respiratory tract. Even it can enter into the human body through broken or ulcerated skin. And then inside the human body, it can transform itself as a cyst or as a trophozoite state according to the condition. If you look at the epidemiology of acanthamoeba, particularly in Southeast Asian countries, so cases have been reported from Thailand, Malaysia, Singapore, Indonesia, as well as Philippines. So now treatment options, modern treatment versus alternative treatment. Like what is wrong with the modern medicine? Why do we need to search for the novel natural products? And how confident we are in order to obtain alternative therapeutic option? And when this dream will come true, usually it takes a long time. There are multiple trial readiness level associated with new drug. So still we are confused which way to go, either modern medicine or alternative. So currently available drugs which are available for acanthamoeba infection includes biguanide and diamidines. But unfortunately, these drugs are associated with major drawbacks such as high toxicity. They have lots of side effects. The efficacy is not same, and then sometimes they have also high cost. And moreover, the patients have to go for the long treatment durations and frequency of administration of these drugs. So what is good in plant-based medicine? Actually, scientists are working on plant-based medicine since last few decades, and according to them, they are the good strategy for finding antimicrobial. And during the last few decades, they have found many compounds which are potential against cancer and against other microbes, including parasites. So moving towards another part, like why did we use, what is that, and why did we implement nanotechnology? What is nanotechnology? Basically, it's a nanoencapsulation of a drug or therapeutic compound within polymeric nanoparticle or metallic or lipid nanoparticle. Basically, it helps in drug delivery, solubility, specificity, and also they help to reduce the side effects. It's a novel approach in delivering biological compound or a drug in a more potent and effective way. So the objective of the study is to synthesize the polymeric nanoparticle and encapsulation of the pure compound into the polymeric nanoparticle, and further down to evaluate the herbicidal activity of prepared novel nanoparticles and its cytotoxic toxicity evaluation. So moving towards methodology, basically polymeric nanoparticles were synthesized, their surface morphology studies were done using SEM and then finally these novel nanoparticles were tested against acanthamoebas, and then cytotoxicities were evaluated. And statistical analysis were done for the final data. Moving towards results and discussion, here are some of the images taken by a scanning electron microscope and ordinary microscope for acanthamoeba trophozoidensis. First picture shows the picture of trophozoid. We can see the visible acanthopodia, whereby second picture shows the distinct shapes of the cyst stage of acanthamoeba. So we can see the different distinct shapes of acanthamoeba cyst stage. So moving towards the chlorogenic acid nanoparticles, so we can see that the morphology and the surface area of chlorogenic nanoparticles, they were found to be spherical in shape. Periglossine nanoparticles were also found to be spherical in shape, whereby gallic acid nanoparticles were found in two different shapes, like sheet shapes and also spherical shapes. So here's a table on the morphological characteristics of chlorogenic acid, gallic acid, and periglossine A nanoparticles. So the maximum size was shown by chlorogenic acid nanoparticle, that was 1 micrometer, whereby gallic acid demonstrated nanoparticle with size of 500 to 800 nanoparticle, and periglossine A nanoparticle were reported to have size of 100 nanometer. So the percent encapsulation were calculated by using this formula, and then the maximum percent encapsulation was reported for chlorogenic nanoparticle, then followed by periglossine A. It shows 91% encapsulation, whereby the gallic acid nanoparticle shows 82.86% of encapsulation. So if moving towards the release of the compound within 24 to 72 hours. So the gallic acid release was 98% within the time of 72 hours, whereby followed by chlorogenic acid, which showed 80%, 85% release, and then periglossine A shows 70% release after 72 hours. So moving toward the amoebicidal activity of nanoparticles toward the cystic stage. So all of the prepared nanoparticle were effective in comparison with the currently available drug, that is our positive control here, chlorhexidine. Similar results were demonstrated against the trophozoite stage of acanthamoeba as well. Among all the nanoparticle, gallic acid shows highly potential against the trophozoite stage. And all of the prepared novel nanoparticles were found to be effective in comparison with the currently available drug, that is chlorhexidine. So moving towards the conclusion, we can see that synthesized novel polymeric nanoparticles showed potential amoebicidal activity with lesser side effects or cytotoxic effect. These polymeric nanoparticles can serve as an alternate therapeutic option in future to treat the infections caused by Acanthamoeba species. And then implementation of nanotechnology can actually reduce the chances of toxicity and also helps in the target specificity and delivery of the drugs. So here are the papers which have been published from the same study and reference of my studies. So thank you so much for your kind attention. Thank you.